Is it working?
Looks good, Jerome. So we can go ahead and start for the day.
Okay. Welcome everyone, wherever you are. It's a pleasure to organize this webinar today. Um, my name is Jerome Lecoq. I'm an investigator at the Allen Institute, and today we are having a big event for us, which is a data release for the OpenScope project. And we have a number of presentations to facilitate collaboration and reuse of those data set. And I'm gonna sort of introduce that a series of talk today. First, some of you might know what the OpenScope program is, but I'm going to sort of speak to it a little bit so that you have a little more context. Before I do that, I want to it's important for me that you know that there's a lot of people behind this OpenScope project, from scientists to engineers and technicians, people that are acquiring the data, as well as the steering committee that drive this project with the support of the NIH and the Allen Institute. So please keep that in mind throughout the talk. This is really teamwork. Okay, so a few years ago, almost a decade ago, Christophe Koch and Clay Reed had this vision of building an observatory of the mind. And so they came to the Allen Institute to make this vision a reality. And so what I would like you to take home today is that we actually make this vision a real assets for our community. The way we did this is by building pipeline that acquire either electrophysiological recording using neural pixel probe or calcium imaging using two-photon imaging instruments. And over the years, we made this pipeline more and more sophisticated, so it has to collect rich data set about the activity of the cortex and subcortical areas. We release those dataset to the community. Those were dataset designed at the Allen Institute for research that we are interested in, but we release it to the community over the years across multiple data releases. And this had an impact. We were sort of pioneer in this space. We were inspired by astronomy, which has shown the impact of shared platform by a group of scientists. So on the left here, you see the number of publications coming from an observatory located in Hawaii. And as the platform was going online, the number of papers generated by the shared platform, the platform used by the community as a whole, broadened. On the right, this is a recent publication put together by George Siegel and Saskia de Vries, which show the number of publications that are reusing the dataset that we collected as part of the Allen Brain Observatory pipeline. But these dataset were designed by the Allen Institute scientists to really make this vision of an observatory a reality. We had to actually open up our platform for external ideas. So this is what we did a few years ago. We essentially started to pilot this program and eventually got it funded by the NIH to allow scientists from across the world to submit projects to a review process and have these selected and released to the community eventually. And today we have one of the first important data release coming from this NIH-funded effort. Our intent is to build a community and help develop the system neuroscience ecosystem. We think we are pioneer in this space, but we want also to help other similar initiatives grow. So this resource has several components. First, you get access, if your project is selected, you get access to this pipeline and can submit project. This project is selected through a double-blinded review process. The next call is this summer. This will be the last call for under NIH funding at the moment. In addition, we have developed a cloud data deployment and an analysis ecosystem. You're going to hear about it today. And we organize community growing events. So today we have a scientific workshop to help disseminate data set, but we also organize a community workshop for people to come at the Institute and learn how to run the experiments. This is an example of pictures here of the events that we hosted in the last few years. There's another one that is happening next June And the deadline is very soon. So if you're interested in learning about how experiments are acquired, you can do this through this workshop. Okay, so throughout this webinar today, we are going to make something different from what most research labs are doing. We're going to release our data prior to publications. So this is a strong effort towards collaboration in neuroscience. And I think it's important to us, and to me in particular, that we set the rules of the game in a very respectful way. And so what I mean by that is that as you are getting access to this data set really openly, think about the team that are behind of this data set. We are sharing with you data set that could push your own research, but I invite people to reach out and build a more collaborative cultural effort in neuroscience. Join forces with those teams. And so to facilitate this, you'll see today that we put a lot of intent in facilitating data reuse. We did this carefully with a lot of documentations and share code. As you have access to these powerful tools, consider joining the Slack channel and collaborating with those teams, so that we all benefit from this new cultural shift in how we do conduct neuroscience experiments. So for a schedule today, we have several talks. 3 talks from each individual team from which we selected the project last year. The first, we have a representative from this team, Jay Pena from Canada. Is part of the Dendritic coupling project, and we are releasing data today from this project. Our process is to have a 1-year embargo for each data set so that the team has the time to push their analysis. In consultation with the team for the Dendritic coupling project, we decided to make the full release in July, which aligns with where they got full access to all of the collected data. The second project was led by Andrew Bastos, Alex Meyer, and Jacob Westerberg. Similarly, we are releasing data for them. They got access to the full data set next week, a year ago. And so our full release will be on February 1st. You'll see you're going to have access to data notebook to access this data set on that date. The third talk is going to be about the resources and the tools that we put together over the last few years to facilitate data reuse in our community. So this will be about the data portal that we are launching today, and notebooks. One thing we call the OpenScope Databook that facilitate a lot of introductory coding onto the dataset we release. And the last talk will be from Yi-Young Shin, which will introduce the Illusion project that she led with Ilal Adisnik. The full data release happened today, so she got access to her experiment a little earlier than the other project. I want to emphasize that I'm really thankful for all of the speakers. Some of them are on the other side of the planet, and so it was a little bit of a challenge to get everyone to speak at the same time. And so as a result, Jacob Westerberg couldn't make it, but he made a pre-recorded video. And so you'll be able to ask questions to other team members, Andrew Bastos and Alex Meyer, today. With that being said, I'm going to hand it over for Jay. We will sort of speak about the first project that we are releasing data from. All right, Jay, are you there?
Yes. Sorry, my screen shifted here. And hopefully you can see my first screen. Great.
Yeah, I'm just going to make one plug before Jay starts. If anyone has questions throughout, please enter them into the Q&A box, and we can moderate those and ask them after each talk. Okay, Jay, if you want to take it away.
Okay, great. Yeah, thanks, Carly, and thanks a lot, Jerome. Just want to say I've been really fortunate to be able to work now on a couple of these OpenScope projects, and I've been able to work a lot with Jerome, and I really, we all really appreciate many of his, very individual and detailed efforts in getting these projects going and getting our analyses to work well, and all the processing involved with them. There's quite a lot of work that goes on behind the scenes that isn't necessarily captured in the final product here. So we're just getting going on the data analysis for the newer project that Jerome was mentioning that we're going to be releasing the data on now. And so it's actually a natural follow-up from our original project. And so I thought we could discuss our original project that we've just been able to get published in terms of the science results recently, and then talk about the follow-up project that we're working on now, and you'll see how they're well connected together. And so in general, what we're interested in is testing predictive learning models and predictive hierarchical learning in mouse visual cortex. So there are lots of people involved in this beyond myself. I just want to go ahead and thank and acknowledge beforehand, especially involving the co-lead labs of Joel Zilberberg, whom I am a postdoc with, and Blake Richards, who's at McGill now, and several of Blake's grad students, Colleen Gillen, Josh, former grad student, I should say, who's now in Imperial, and a new grad student, Josh Tyndall, in his lab, and a PhD student that I've been working with in Joel's lab as well, Tim, and lots of more collaborators, including lots of Allen collaborators, especially Jerome, Sheila, and Natalia that we've worked quite closely with. So in this first set, we'll look at, in the first part of the talk, we'll look at this first project where we looked at how pattern violating stimuli drove responses in somatic and distal apical dendritic compartments, and how those responses evolved differently from each other over days that drove this follow-up work that we're working on now. If you're interested, we have an early release for now in Journal of Neuroscience, and also a full data set description in Scientific Data, along with repository linked there. Okay, and so we want to know whether the visual system implements a predictive hierarchical model of the world. These sorts of models have been around for quite a while, dating back to the 90s with Helmholtz machines and the seminal model of Rao and Ballard in '99. And one of the really appealing parts that has brought them more and more to the fore is that they seem to address in a natural way many of the implausible aspects of feed-forward networks that people have noticed from the very beginning when backprop was first introduced. And of course, within machine learning, many of these issues at a more pragmatic level are being addressed as well. But for predictive, so this is one class that of model that seeks to address some of these issues. For example, predictive hierarchical networks can be self-supervised, and instead of requiring the so-called weight transport involved in backpropagation, they can learn through more local Hebbian types of learning. They naturally incorporate dynamics, and some more recent theoretical work has shown how it can reduce to backprop in certain limits. And there are, in fact, more advantages. So I encourage you to look more into it if you're interested. There are lots of implementations of this general idea that have come out over the years. But the general idea is that higher order brain areas make predictions about incoming stimulus data, where they can be compared, and then the differences between those predictions and the actual stimuli can then drive learning in the internal model that's generally thought of to be encoded in the synaptic weights. Okay, so in particular, that means unexpected features, novel features of stimuli can drive learning. Okay, so in the first project, we tested this by looking in just a single brain area. And we could do so by taking advantage of some of the interesting anatomy involved in pyramidal neurons, especially in L5, but also in L2-3, though in part due to somewhat different, potentially biophysical reasons. So the idea is that distal apical dendrites in the pyramidal neurons in layer 1 tend to receive lots of top-down inputs that might be related to, for example, these top-down predictions. Whereas the basal dendrites that are very close to the somas receive bottom-up inputs that the soma tends to then reflect by integrating those inputs together. But what's important is that those two compartments, the apical dendrites and the somas, tend to be electrotonically segregated, either by distance or potentially through other mechanisms, other gating mechanisms like inhibitory interneuron activity. Okay, so just as a quick walkthrough for how we then set up our experiment, we had the mouse look at a screen in the full visual field, introduce these visual stimuli. The main one that we'll talk about is these sequences of Gabor patches that were adapted from other similar work that we had seen that seemed to potentially drive some of the responses that we're interested in. And in particular, we had two sets of sequences, an expected one or a sort of regular sequence here that comprised of 4 image frames, A, B, C, D, followed by a gray screen. Each image frame had the Gabor patches located in different places. And then later on, we violated those patterns on a different day with a new image frame where those image patches were orthogonal to the ones that they would have expected in the regular sequences. And it turns out to be a pretty nuanced stimulus. So the total sequence length is only 1.5 seconds. So I'm going to show an example here. What you'll see is a green dot in the lower right-hand corner that indicates that we're in the regular sequence, in the regular sequences, A, B, C, D, and then gray. And then a red dot that will indicate that you should start seeing by the 4th frame the orthogonal patches. The mouse wouldn't have seen the green or red dots. It's just a visual cue for us because, again, these go by quite quick. It's only 300 milliseconds per image frame. And so once we get to the red dot, we'll see that 4th image frame be orthogonal in the mean orientation to the image, to the patches in the preceding frames. Okay, and while the animal was seeing this, again, full visual field, we also recorded their running velocity and pupil position and diameter using deep lab cut in order to track the pupil, in this case over here. Now, in our case, we use this in order to make sure that any activity that we saw didn't arise simply from behavioral considerations, because in rather seminal work by Neal and Stryker, they showed that indeed both of these behaviors seem to lead to increased activity in visual cortex. And so we wanted to make sure that what we saw was more likely to be related to top-down inputs rather than behavioral changes. Okay, and then of course we imaged the 4 different planes, and we can see a quick example of these over here, sped up. And I want to note these are all taken in different animals. They're not simultaneously imaged, but that's what we address in our follow-up work that we'll see. Okay, and so just with a little more detail, the habituation occurred over a period of 11 days, with 6 days where they were exposed to the regular sequences, A, B, C, D, and gray, or to the other visual stimulus, the same regular patterns that I'm not showing here. If you're interested, you can follow up with the paper that I was mentioning at the beginning. Bottom line is it basically worked as a bit of a control stimulus against which we could compare the results that we saw in the Gabor stimulus case. And then finally, we recorded these the neuronal activities over a period of 3 days, where in those 3 days, the unpredictable sequences occurred in about 5 to 7% of the stimuli. Okay, so we, as we mentioned, imaged the 4 different planes, and we're interested in activity that might be associated with those predicted by predictive hierarchical networks, such as error signals, for example. Importantly, we needed, therefore, to not just look at the dendritic and somatic activity, but to be able to match the cells and the dendrites across days in order to get a better feeling for whether that activity is driven by the same neurons, or if we sample different neurons, then that could confound any results that we end up with. And so we were able to adapt the matching algorithm from the Allen in order to do similarly for the dendrites, as you can see here. Okay. And then we saw what sorts of activity this resulted in. And so in the very first day, each session is taken on a different day. We see that the dendrites, for example, already have, for in L23, a difference between the regular sequences that we show here in gray, so that corresponds to the ABCD frames, and the unexpected U frames that correspond to the colored plot, to the colored contours. Similarly, in the somas, we see differences in the responses to the D and U frames. And we then looked at that over a period of days. This is all of the ROIs right now, averaged together. And you can see that a couple of things. First of all, the somatic and dendritic responses seem to be a little bit different. And the evolution between those two is a little bit different. And so if we then look at the mean difference between these two curves, between the unexpected and the expected curves, then we get plots like we see over here, where we can track those differences over all 3 days for all 4 imaging planes. And again, intriguingly, they're different animals, but we see that those differences seem to increase in the dendrites and decrease in at least some of the somas, or at least not increase. We'll look at this in the tracked ROIs here in a moment. Okay, so in order to get a better feeling for how those responses change for the same neuronal elements, we look here at a similar plot where each line corresponds to a measure of sensitivity of the neuronal element to the pattern violations, and we measured that by something we call the USI, which indicates the sensitivity to the unmatched patterns, to the ones that do not match the regular sequences. Okay, so we took the difference between the means and then normalized them based on their variances. And so this is basically a signed version of the so-called sensitivity index, or D prime measure, that you see a lot of times in behavior in order to see whether or not a signal is detected. Okay, and so overall, what we saw here was, again, different patterns in the dendrites and somas, again, different animals, but intriguingly, the dendritic activity seemed to overall increase, whereas the somatic activity seemed to decrease towards 0. And we could see this if we look at, for example, the absolute value of the mean USI activity over days, which increases for dendrites, decreases for somas. And then if we look at the variance change as well, we can see that in particular, the somatic activity really collapses down towards 0 and the dendritic activity does not. And this now comes to bear on some of the ideas in predictive coding networks that we were seeking to test. So, for example, in some theories, the error is encoded in the apical dendrites. And if that were the case, then over the course of learning, you might expect such a learning signal to decrease towards 0. And so the difference between the unexpected and expected activity should go towards 0. But in fact, we don't see that in the dendrites. We do see that somewhat in the somas. And so this seems to be perhaps more consistent with more classical models of predictive coding. So, for example, Round-Ballard, than it does with some more recent work that suggests that error signal is being computed in the apical dendrite. We then also were able to look at how the sensitivity on one day predicted the sensitivity of that same neuronal element on the next day. What we found was, in the case of the dendrites, from session 1 to session 2, they changed their preference from either the pattern violations to the regular sequences or vice versa. So if they were more sensitive to the unexpected or the pattern violation sequences, then they were more likely to change that sign and become more sensitive to the regular sequences or vice versa. We looked at this more carefully. It turns out some correlation results not related to the ones that you see right here, you have to be careful about. There's some bias associated with it. And so one fun outcome of our analysis here, when we looked more carefully at the same sorts that I'm not going to show right now for the visual flow stimulus that I mentioned. So it turns out that in some cases, these correlation measures can be biased. There's a problem known as regression dilution. And one result of this was that we were able to develop a method to correct for that bias and give even more insight into the responses that we observed in the other visual stimulus scenario. Okay, and so I mentioned that it looks like the activity diverges between the compartments, again, in different mice, but the dendritic activity seems to decrease, or the differences between the responses from the pattern violation to regular sequences seem to increase for the dendrites and decrease or stay the same for the somas, and decrease overall in magnitude for the tracked somas, as we saw. We looked at this a little more carefully by piecing apart the activity between the unexpected U and G frames that we see in color and the regular sequences, the ABC, not the D. That's a bit of a more involved Gabor frame. And what we saw was that this divergence only occurred between the compartments, between the dendrites and the somas, in the case of the pattern violation sequences. But with the regular sequences, in both cases, the gray, they seem to increase or decrease more or less together. There doesn't seem to be this divergence, suggesting that this might be a bit of a more general pattern that we might see if we looked at more simultaneous imaging in individual mice. And so that's what we then have been working to look at in our current OpenScope project that we'll be releasing some data for right now. In particular, how this dendrosomatic coupling in the context of sensory prediction might look like and change in mouse visual cortex. And there are, in particular, two hypotheses that have been proposed that we are looking to test. And I want to mention, even if that data that I just showed were taken in the same animals simultaneously, there are different ways to measure coupling, and so that might be a little bit of a different way to look at it compared to more classical ways. In more classical ways, you look at the activity between the dendrite and the soma, and you just look at, for example, measures of correlation in those activities to measure the coupling strength between the apical dendrites and the somas. And so in this first hypothesis, the idea is, well, you're only going to get large enough signals that propagate from the distal apical dendrite down to the soma in the case where you have also near simultaneous or corresponding activity in the soma, resulting in back propagation of that action potential that then allows for the activity to pass from the apical dendrite. And so in that case, what you would expect are lower correlations when the predictions don't match the incoming sensory data, and higher correlations otherwise. And so that those correlations should increase as the animal gains experience with the stimulus and is able to make better predictions. In contrast, there's a different hypothesis that, like I mentioned before, supposes that there are error computations that are either exist or that occur or are co-located in the apical dendrites. And when those error signals are sufficiently strong, in the case where they're mismatched with the incoming sensory data, then you can get a plateau potential that couples the activity between the dendrite and the somata together. And otherwise, in the case of matching, then you won't get such correspondence of activity. And so in this case, higher correlations would be expected for the pattern violations, and then that coupling would decrease over days. So these are really opposing hypotheses that that we can now test, and we can test it specifically by looking at near simultaneous imaging, which we'll look here in a second. And so the stimuli that we look at are again the sorts of Gabor stimuli that we saw before, and we've added a bit more there to it as well. But now we'll also look at naturalistic movies where we can reverse the flow, violating the expectations that they're habituated to. And so here are some movie clips where you can see that. We use 3 different movie clips. We habituate them to just the forward direction. They're 9 seconds long each, and then we reverse them during the experimental sessions. Here's another one with conspecifics with 2 mice. And again, you can see the direction flow right there as the mouse runs backwards. So that we would expect that to potentially violate the expectations. You don't oftentimes see mice running backwards, for example. Same thing with a person walking backwards in the previous one. And then finally, some earthworms that they might prey upon, for example. In this case, this earthworm falls and now pops back up, as you can see. And that might violate models of the physical world that they might have, for example, that even perhaps coarsely or statistically reflect effects of gravity. Okay, and so so we need to really simultaneously image and then couple together the ROIs in those simultaneous imaging between the dendrites and the somata. And since we want to look to see how this coupling evolves over days, not just once they're exposed to the unexpected stimuli, we also, in this case, image in the final 3 days of habituation when they're only exposed to the regular patterns. So in the previous experiment, we only looked at primary visual cortex, but in this experiment, we can also look to see the correspondence of these sorts of computations across the visual hierarchy, including in LM, PM, and AM. And all of these are near simultaneously recorded at around 10 hertz. And I should mention there's some, with the multiplane mesoscope, there's some issues of ghosting that can occur. So this is another place where Jerome's been super helpful as we've been working to correct for those problems, and they've been working on the same at the Allen. So so we can get some really good, reliable results here. Okay, to be able to do so, we really need to track to connect the dendrites to the somas. Unfortunately, a classical sort of Z-stack doesn't work very well. So this is the sort of Z-stack we would have attained before in the first OpenScope. And so we worked with the Allen, especially with Natalia and Sheila and others there, to develop a new protocol to get some high resolution Z-stacks. They're basically mini movies for each plane. PhD student Tim Henley, that I mentioned before, has been working on a lot of post-processing techniques involving removing independent noise, really emphasizing local structure, correcting for intra and interplanar motion, combining the multiple stacks together, and then finally matching the imaging planes to the Z-stack planes. And so the resulting Z-stack, as you'll see, is much cleaner, much nicer. But there's still challenges with tracing even these much nicer and high resolution Z-stacks. Here's an example where we did some automated tracing and some nice preliminary results, but there's also a lot of sorts of traces that aren't so reliable. And so Josh Tyndall and Tim have been working to more carefully trace these somas, and here's an example of some preliminary results that they've been getting there. So we're really excited to then be able to take that to the activity of the dendrites and somas and see how that coupling varies with context. And so overall, we found in the first project that indeed we found activity that was consistent with some, but not all, predictive learning models, and that these bottom-up and top-down signals evolved differently and might suggest a context-dependent dendrosomatic coupling. Some analysis challenges led to a new statistical method that we'll be putting up on bioRxiv here in a moment that can be applied to a lot of other neuroscientific data, as it turns out, and we'll give some examples there. And we are now currently following up to examine this context-dependent dendrosomatic coupling in more detail across the visual hierarchy with near simultaneous imaging of the distal apical dendrites and stomata, along when they're exposed not only to the artificial Gabor stimuli, but more naturalistic movies as well, and then coupling them anatomically together with these high resolution Z-stacks. And so please, like Jerome was saying, love to work with people. Feel free to reach out if you're interested in this data set, including our emails here for Joel, Blake, myself, and the Slack link that Jerome has mentioned. So thank you very much.
Thank you, Jay. Just a reminder for participants, you're free to ask questions in the Q&A box, either for Jay, any of our awardees, or on OpenScope in general. And I encourage the presenters too, if they have questions for each other, feel free to speak up. And we'll talk about how to access data for each project in a bit. Jerome, anything to add to that before we can move on?
I'm curious about, because I know with Colleen, you've released some data set already about a year ago. We did that together. And now we are pushing more data out. And so what kind of opportunities do you see between these two analyses that could be done? Because I'm sure the newer data set is going to be richer in many dimensions. But the older data set is much more finalized. You have finalized the notebook, the figures. And so I think for external people to use, it's probably easier to start there. And so I was curious if you had some guidance for people to get started on the previous data set on things that you think would be beneficial to your work.
That's an interesting question. One thing that we're excited about with the new data that, of course, we've been again getting big assist from you on is applying this independent noise remover, this deep interpolation model to the movies to be able to get much cleaner data. So in the previous data set, we weren't able to apply that and then segment properly. We've been able to Tim's been able to adjust our segmentation algorithm to be able to get nice dendritic, ROI segmentation with the cleaner data. And so with the older one, that may have specifically affected some analyses that we were interested in, where we wanted to look at intra-session plasticity changes that might result. And we did find some, and again, I'll refer you to the paper to take a look at that. It's not clear, how sort of whether there was nice interpretable systematic changes that we saw in the session. Those were more relegated to the cross-day sessions. So I think, one thing I would be really interested in is applying the deep interpolation model that you've made available to the previous movies, for example, resegment those movies, and that might remove enough noise to reveal some more interesting and systematic short-term plasticity changes that are in those data that we were only able to get some hints of in the first one. And the nice part about the first project is that the acquisition rate is greater as well. And so that might be a better, in fact, project to address some of those questions with by doing that.
That's a good idea. I do want to mention to everyone that for this project, we actually released the raw data. So these are actually quite large files. And as you said, there have been opportunities to develop new, and actually I know for a fact that some external groups have used those raw data files to develop their own methodology on top of it.
Okay, Jay, thank you for that overview. If there's no other questions, we can move on to the next OpenScope awardee, and we have a recording from Jake Westerberg, but we also have teammates Andre Bastos and Alex Meyer here to answer any questions that come up on the talk. I'm going to go ahead and screen share the recording. Alex and Andre, please stop me if there's anything you want to introduce before I start the recording. Otherwise, I'll go ahead and screen share. Everyone see that?
It's great.
Hey, I'm Jake Westerberg, and on behalf of our OpenScope team, I'm excited to give you an overview of a NeuroPixels OpenScope dataset designed to investigate the neural underpinnings for predictive processing. Specifically, we were interested in understanding how specific oddball stimuli are detected across cortical circuits. Now, I apologize for not being there live with you. The time zone difference from Amsterdam is a bit prohibitive in that regard. Nonetheless, I hope this presentation will convey some of the critical aspects of the project. For one, I will provide you with some of the background to the project and why we think this is an interesting avenue to explore. I will follow that up with a description of the experimental design, what conditions are present, and the different opportunities for comparisons. I will go a bit into the quality and scope of the data as well. And hopefully I will get a chance to show you some of the interesting preliminary results and detail to you what you might expect to see should you start looking into the data yourself. Lastly, I will conclude with some ideas for opportunities that could be explored and hopefully inspire you to come up with some of your own ideas. I will also say this now and again at the end. We're excited to have some of you reach out to us. We're always looking for enthusiastic collaborators, and we're happy to help to answer any of the questions that you might come up with as you start exploring the data yourself. And now, without any further ado, as I mentioned, the project is designed to investigate predictive processing. And to accomplish this, we used temporal oddball task where certain stimuli defy expectations. This task, developed about 15 years ago, habituated participants to a sequence of auditory stimuli, but the same can be done with visual stimuli. Now I will focus on the bottom half of this plot because it is most related to what we ended up doing with the mice in our study. Essentially, participants would repeatedly be exposed to a sequence of stimuli where the last was saliently different from the preceding stimuli. This makes that last stimulus a local oddball because it defies its local context. As cartoonified here, the local oddball is the red stimulus with the standard stimuli denoted in green. After habituation, in the experimental block per se, the participant would mostly experience the same local oddball sequence. However, on a small number of trials, the local oddball would be replaced with the same stimulus as those preceding it, no longer making it a local oddball. However, it does still defy expectations, and we call this a global oddball. Between local and global oddballs, we can investigate how two forms of predictive processing are accomplished. Indeed, the investigators in this study documented notable changes in the EEG event-related potentials associated with the different oddball types. And since then, there has been quite some work to understand the neural basis of these detections, something that we believe that this data set that I will introduce to you now is quite well prepared to do. Okay, now we thought to situate this question within a predictive coding framework, specifically a variant that uses functional differences between cortical layers and hierarchical processing to accomplish such oddball detections. That is, we expect to see differences in detections as a function of cortical layer and position of a given area within the cortical processing hierarchy. Also, we suspected that specific inhibitory cell types might be at the heart of predictive processing. Really beautiful prior work from a few years back showed that certain cell types, parvalbumin and somatostatin positive interneurons specifically, change their population firing with sequence learning over subsequent days. Given the opportunity to interrogate specific cell types through optogenetic tagging, we sought to experimentally determine the role of these somatostatin and parvalbumin cell types in oddball detection in our task. Okay, now to dive into the design of this experiment. First to note is that we have two cohorts of mice with respect to the stimuli that they experience. That is because each mouse was habituated for about 10 days to a specific sequence of full-screen oriented drifting bars. One sequence had leftward bars as the standard and rightward bars as the local oddball, and vice versa for the other cohort. It is important to note that the mice are passively viewing these sequences. They're not engaging in an active task. Each sequence consisted of 4 stimuli presentations. In one cohort, they were trained to left, 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 right, for example. The dataset consists of 16 mice in total, divided up as seen here. After the roughly 10 days of habituation, mice were prepped for the NeuroPixels recording session. In that session, the mice first experienced the same habituated sequence for 50 trials. Please do note that up to this point, the mice still have never experienced a global oddball sequence. We did this to enforce the local oddball predictability as strongly as possible, to maximize recognition of the global oddball as such during the recording session. Then, the mice moved into the experimental phase. On the majority of trials, the mice experienced the local oddball sequence, but on 20% of trials, they experienced the global oddball. I have the timings here for reference. Stimuli were presented for 500 milliseconds with 500 milliseconds between presentations. After the experimental block is finished, we run a few other block types for controls and comparisons. First, we have randomized sequences, which consisted of randomly chosen patterns of the same 2 stimuli. For instance, there will be sequences of left, right, or right, 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 and so on. This can be used for orientation tuning, for instance. Next, we have an adaptation control, where the mouse is exposed to alternating 4 stimulus sequences of the 2 stimuli. It is perhaps the simplest block, just alternating between the 2 sequences, those being left, 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 right, 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 left, 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 and so on. This has the global oddball sequence from the experimental block, but in a different context for an interesting comparison. We then run a shortened block of the experimental phase again for a number of reasons, including to observe changes in firing rates across the recording session. And lastly, we have receptive field mapping and optotagging, which I will go into a bit more detail about later. Okay, now while the mice are experiencing those conditions, we have NeuroPixels recordings from 6 primary cortical targets: V1, LM, RL, AL, AM, and PM, as well as the thalamic nucleus LP. This gives us a nice set of laminar recordings of areas spanning the visual cortical hierarchy through which to test our hypotheses. It is also worth noting that there is substantial data from other brain areas, more than what I can list here. So if you have a different area in mind that you think might be interesting to explore in this task context, you might just find it in the complete data set. Okay, now to give you a sense of the data, we have validated that you can expect that the quality of single neurons in this data set is comparable to that of the previous release of Allen Institute collected NeuroPixels data. Here you can see in green the counts of units for specific unit metrics after applying the same criteria as used previously. Specifically, the quality metrics were drawn from this Nature paper from a few years back. Without going into too much detail, you can expect a great deal of high-quality data, and if you're just interested in bolstering your own set of data for exploring spike waveform properties, for example, you can of course grab that information from this data set without even having to interact with the electrophysiology. Okay, here I have numbers of cells broken down by mice and cell types. Most mice include data from all target areas. We get fairly consistent sampling across animals, and I will have the exact summary numbers a bit later on. I also have the summary of the high-quality PV and SOM cells here on the right. There are about 1,000 units of each type in the high-quality data, and more in the broader data set should you want to focus your efforts on those particular cell types. Now, to give you a better sense of the optotagging data, I've divided out the cells deemed to be optotagged using the criteria in the bottom right, and show them in each of the optotagging conditions. Each of the panels is a different combination of stimulation waveform, strength of stimulation, and cell type. Firing rate is on the y-axis and time relative on the x. To briefly summarize, we stimulated these at 3 strengths with 3 waveforms, as detailed in the bottom left. Beyond being used for identifying cell types, we do have more stimulation patterns than previous datasets. Even this aspect of the data could be used to investigate properties of these cell types with respect to stimulation patterns, a yet unexplored opportunity in this dataset. Now, to prepare for some of the data from the experimental block. I have again here the target brain areas that you will see data from. As a bit of a sanity check, we can ensure that the data are as expected to a certain degree by looking at visual response latencies, as depicted here. These should follow the hierarchy of areas, which they seemingly do, as color-coded. In that central plot, you are seeing the average responses of each area in time relative to the start of the visual stimulus presentation. At this point in the population analyses, you will notice that we have started normalizing the data, hence the arbitrary units. Here are the summary numbers for the data I will show you. I want to make it clear that the complete dataset will have the roughly 30,000 units included, but I have the restricted set here for our analyses. Much like the unit quality metrics, these follow the numbers of previous datasets quite closely. And as for how we'll be looking at the data, given the time and that I wanted to focus more on giving you an overview of the data rather than a scientific report, I will be primarily sharing the population level analyses of the single unit data. And we'll be primarily comparing the responses to oddballs in the experimental block to responses in the adaptation control block. Moreover, I will be doing a sort of local normalization by subtracting the responses of neurons to the third stimulus in the sequence from the response to the oddball or the oddball comparison response in the control. In the plot on the right here, it is subtracting B from A and D from C, and comparing those differences for specific oddball types. These plots on the right show the population average responses to these sequences, but as you will see on the next several slides, as we do this local normalization, responses will look a bit different. Primarily, instead of seeing almost universally positive responses, you will see that the responses sort of vary around 0, because in certain conditions, like in the adaptation control block, we expect the responses to the fourth stimulus to go down relative to the third, whereas in the oddball detection paradigm, we might expect those responses to be increased and not decreased. But before we get to that, while I won't have any examples of LFP responses beyond the one shown here, I think it is important to note that there is an entire complement of LFP data included in the dataset. We have only begun to explore it, and I am sure there is a lot for you to find once you start digging in there. Here in particular is an LFP response of a single mouse in primary visual cortex averaged over the global oddball sequences presented and plotted with depth on the y-axis, time on the x, and the LFP polarization indicated by the color. Okay, now to look at the population level responses with respect to brain area and oddball type. In each of these panels, you are seeing 2 of the responses. Magnitude is on the y-axis and time relative to the stimulus onset on the x-axis for each. Each pair of plots is from a different brain area. Higher order areas on the left and lower order on the right with the thalamic nucleus. Gray clouds are the 95% confidence interval across units for the control comparison, and the colored clouds represent the oddball signature. Statistics were performed and indicated by the black bar if it is present for a given plot. The number indicates when in time there is a significant difference between the clouds. We find robust encoding of the oddball stimulus occurring quite early in the visual response and already at the earliest areas in the processing hierarchy for the local oddball. Global oddballs are more weakly encoded, it seems, and only certain areas seemingly show a robust signature. LM in the upper right in particular seems to, at least relatively, respond quite robustly to the global oddball. LP in the bottom right doesn't show as pronounced oddball responses. So to just briefly summarize, we see that there are signatures of oddball detection, both local and global in this data set, with perhaps some quite interesting differences in how each of these types are detected. Aside from population level dynamics, we do see that there are individual units that seem to be global oddball detectors. I don't show local oddball detectors here, but I can assure you that the dataset is filled with them. Individual unit global oddball detectors are more rare and therefore perhaps more interesting to identify. These examples each come from different mice and show spike rate as a function of time for 2 conditions. I will note that from these examples, it does look more like a global oddball detection comes later in the response when looking at the local context alone. What I mean by that is that the red global oddball response here is compared to the preceding stimulus in the same sequence, not to a different control block like in the previous plots. I will also note that these are all enhanced responses. That is important because you would normally expect decreased responses with what would otherwise be an adapted stimulus. And while global oddball detection responses are seemingly much weaker, they can still be detected at the individual unit level. And this wasn't a given when we went into the project, as there was a lot of discrepancy in reports on whether or not individual neurons could respond to global oddball stimuli, especially in sensory cortical areas like visual cortex. Now back to the population level analysis examples. Much like the cross-area comparisons, we do see some differences as a function of layer in which the data was recorded. Here you see the data aggregated into the 3 major laminar compartments. The upper layers being primarily composed of layers 2 and 3 units, middle layers being layer 4, and deep being layers 5, 6A, and 6B. Again, you'll see the statistics, and one interesting tidbit from this plot is that we do notice that for the global oddball stimuli, the detection happens, or seemingly happens, earlier in the upper and deep layers relative to the middle layers. And this is quite interesting in the sort of laminar predictive coding context, because those are the primary feedback recipient layers of cortex. And so it might be the case that global oddball detection in visual cortex is inherited from higher order areas, perhaps even areas in the mouse frontal cortex that we were unable to record in this particular data set. And much like all the other local oddball plots, you'll see that local oddball detection again happens quite early and quite robustly across all the layers. And as for a last bit of the preliminary data, again at the same population level, it seems that there are both observable differences for both inhibitory interneuron populations in the oddball types. And before we do wrap up, I want to highlight that we understand there are several other comparisons that could serve to determine oddball selectivity. This population metric is just one of many that I could share today. At the very least, I hope you can appreciate that there are some interesting dynamics ongoing across visual areas with respect to these different oddball types. And in my opinion, it's a very rich data set to explore. As one last little thing I want to highlight, if you notice on the right in the local oddball plots, you'll notice that sort of oscillation that you probably were a bit surprised by, or maybe not surprised by, in the previous plots as well. This appears to be some sort of 5 Hz oscillation that has nothing to do, or at least is independent of, the actual physical stimulus. And in talking with others who have been in and around mouse recordings and visual cortex during tasks that might be similar to this, that 5 Hz oscillation is by no means unique. And so that's something you could potentially explore within this data set, being the oscillatory components that contribute to these sort of visual stimulus types and detections. And so I hope that gave you a sense of the data included here. I hope it inspired some of your own questions, and I hope in the experimental design here, you discover questions that we haven't even thought to ask or that have even crossed our minds. Between all the different blocks and stimulus combinations and sequence patterns, we do think there is ample opportunity to discover new and exciting things beyond principles of predictive processing. easily going into adaptation, visual processing, and even properties of visual neuron responsiveness, such as through the data with spike waveform metrics and the optotagging of certain cell types. But before we conclude, I wanted to end by motivating thought and effort in other directions with this data. We really believe that this dataset is full of interesting results waiting to be found, reaching far beyond the specific questions and hypotheses I have focused on here. A few things immediately came to mind when I was preparing this. One is that this data can be used to investigate propagation and adaptation of signals through cortical microcircuits. This seems to be a great deal of opportunity to explore principles of laminar function. You can check out this recent publication from our group and others to perhaps get some inspiration. It really delves into differences and similarities between laminar function across species. Another idea lies in understanding how local oddballs are detected and robustly represented as they ascend the hierarchy. We know from previous work like this here that local oddballs have a great deal of power over our attention. And while this dataset is during passive viewing, behavioral measures that I have not even gotten the chance to mention are included in the dataset. Those relevant to my point about attention and this direction in particular could include pupillometry and running speed. And lastly, there are many other options out there that I keep alluding to that I hope you can discover and ask your own questions with. We are truly excited to see what you can pull out of this dataset. And on that note, please do reach out to us if you're interested in the data. We are happy to facilitate your efforts and look forward to what other curious investigators can glean from this dataset. Thank you for taking the time to attend, and we hope that you find us as enthusiastic collaborators in your efforts with this OpenScope project.
All right, so we again have time for questions, and if Alex and Andre want to add anything else or comment on Jake's presentation, please chime in. And again, if there's any questions from the presenters to other presenters, we welcome that as well. Just an FYI, next up we have an overview of the OpenScope Databook by Carter and Ahad here at the Allen Institute.
Carly?
Yes.
I have a question, but I mean, Jake did just such a fantastic job highlighting all the opportunities with his data set. I'm so thankful for that. One point that he could not nail is we just had a presentation from Jason about the presence of unexpected responses in dendrites, and the NeuroPixel dataset might be sensitive to the similar process somehow. And so I'm curious if you think that could face an opportunity for doing analysis across those two, because they have similar experimental design components. I wonder if this is a place where people could also dive in.
Do you want to comment on that, Alex? Well, no, I mean, it is an interesting question there. I guess we should also think about whether or not the NeuroPixels data is reflecting mostly spiking in the soma or also potentially spiking near the dendrites, right? So that's perhaps a question that I would want to address before trying to do that integration. But it does, from basic electrophysiology, we would assume that most of the spikes are centered near to the soma, right?
Yeah.
So if our data ends up supporting more the view that is based on the somatic recordings, where you have actually the opportunity to separate them, then that would be potentially a converging response. But we do have the capacity to look at current source density analysis, which can give us access potentially to some dendritic computations, although it's of course less clear-cut than with imaging the dendrites directly. But that could be a very interesting avenue to explore.
The second question I have is, Jacob mentioned other areas. and in particular the hippocampus, we are running currently a project on the hippocampus about understanding longer scale sequence and temporal structure. Do you have currently analysis going on in the hippocampus? Do you think that's a good avenue for people to go in there and collaborate with you?
Yeah, I think every single probe, or almost every single probe, has data from the hippocampus. It seems like in the mouse brain, at least, you can't ever escape the hippocampus. And I think there's perhaps more data in the hippocampus than there is in cortex. We have to look up the numbers one more time to be sure. And of course, that's where we think these predictive models, one of the areas where they potentially are stored in the brain, right? Especially longer term models that can be learned over several sessions. As our task design enabled, those models may definitely include the hippocampus. So we are starting to look at that with LFP recordings initially. And we don't yet have a strong result there, but certainly that's one of the things that we hope to explore, and we hope to, open up for others to explore as well.
Yes, just so that people know, next year we release actually a hippocampal data. So I think that's an opportunity to sort of grow all of this complementary analysis in a very interesting ways.
Is that data that is similar in nature to these current projects, but with a question more designed around theories and models of hippocampal function?
So the experimental design is tailored for we're trying to find visual stimuli that drive the hippocampus more strongly. So there's no component of surprise as in yours. We'll have an introduction next year on it, but I think there also will be an opportunity there for complementary analysis.
Yeah, it may be the case that these grading stimuli do not drive hippocampal neurons very much. That appears to be the case from our initial, exploration of the data. And on the other hand, it may be that those neurons are more sensitive to, change in context rather than change in the stimulus properties. So if that's the case, that could be something that we could explore. And we're very curious about that, of course.
Yes.
Yeah.
Again, so I just want to emphasize, we said it, we created a Slack channel, I said it earlier, where everybody can join in and start a conversation directly with Andre and Alex and Jacob to sort of have concerted analysis and consolidated efforts. So feel free to go there. I think maybe we should move on to the next talk.
Yeah, so next up we have Ahad and Carter here at the Allen Institute talking about the OpenScope Databook. I will let them take it away.
Yeah, sounds good. Hello, my name's Ahad. I'm a software engineer 1 at the Allen Institute. I've been working for the last year on creating our processing pipeline packaging pipeline rather, to take all our process data and turn it into NWB files that you folks can look at. And I'm going to give a short talk on that, what NWB files are, how you can use them, and then Carter's going to talk about how to access them. All right. Okay. All right, and here we go. So this is an introduction to our NWB files and why and how to use the NWB ecosystem. So your first question is probably, what is the NWB ecosystem? And it's a neurophysiology standard, which is designed to be unified and open source. It's flexible and supports various data types, so extracellular ephys and optical physiology. It allows us to store different stimuli, modalities, different methods of acquisition, processing, raw information, and more. And it's created and supported by the Kavli Foundation and NIH Brain Initiative, which means it's pretty well documented and supported with software libraries. And I've got this little diagram right here that shows how it's called Neurodata Without Borders, by the way. I think I forgot to mention that earlier. And it's just showing how there's different elements to each file that are all stored and shared in a standardized way. And we're going to go into a little bit more detail related to that in just a moment. So what are the benefits of using these NWB files? Well, first of all, there are Python and MATLAB libraries for development, analysis, sharing, and more. So that just means that you don't have to do a lot of software writing if you just want to open a file and look at it. There's tons of libraries that are already designed to do this. You can make edits and save it in your preferred language. Also, there's some visualization tools like HDF View and NWB widgets, which don't require any programming and let you look at things like plots of your data, the eye tracking of a mouse, the probes within the brain, the CCF registration, all sorts of stuff without having to do any real coding. You can just open up a notebook with 1 or 2 lines, enter your file, and look at things like that. There's also a great ecosystem to check file validity. So NWB Inspector can find both errors in your files and best practice violations. So if you're downloading one of our files, doing some edits, and then trying to upload it, you can make sure that you're following the standard. And speaking of that standard, there's a well-documented and evolving schema that just tells you, hey, if you're going to store images, store them in this way. If you're going to store acquisition details, store it here, etc. And then there's great software support on both GitHub and Slack. And just as you can see, these are all links. So if we share the slides later, you'll be able to click on them and access the resources for all these things. And then I've got a little picture here on the side that shows an NWB file. And you can see how it's split up with acquisition here and then eye tracking information. And we'll go into a little more detail there later. And then this is one of those visualization tools, NWB widgets, where you can open this NWB file and see some metadata related to the actual file. And then you can see, if you wanted to, you could click like acquisition or stimulus or modules and see that related information. Now, here is a slide on NWB files and their schema, just to talk about some of the standardization for it. So even though the NWB files are quite standardized, there's a lot of flexibility to help with storing information related to your experiment in the way that you want. And we're going to go over some sections of the file in just a moment, and I'll talk about why that's important. So first of all, there's an acquisition field, which lets you store information about your experimental acquisition. So in our case, we've got stuff like eye tracking, some raw data, some wheel signals here. There's also an analysis field, which we're currently not using, but if you decide to download one of our files and do some analysis, you could store it here. So that way you could share your files and have people with just one singular file be able to look at analysis you've done as well. There's also a general field where we can store experiment and modality specific information. So stuff like device information, your institution, keywords, some metadata related to perhaps how the experiment was done. And then there's an intervals field, which we'll go over in just a little bit, which is dealing with some of the images or movies or whatever that you may be presenting during your stimuli. And you can split these up in different ways if you wanted to perhaps download our file and change something, or download our file and run something similar for your own files. Also, the data types and groups can be used to organize files. So you can take a file and say, hey, I want all the ones that have eye tracking, or I want to look at all the ones that have this plane and stuff like that. Also, there's optogenetic series, stim tables, and more that are all supported by NWB widgets, which is just really helpful for visualizing them. And if you wanted to, for example, plot all of them, you can do so without having to write much code. I'm going to give you a demo of 2 files now. One will be on EFIS and one will be an OFIS file, and we're just going to go through them. These are files that are very similar to the ones that you're able to download after this session is done. So here is the first one. This is a ECEFIS file. So you can see we have like a subject ID, which is just the mouse number, a session ID, which is just the associated session with the mouse, and then it just says like optogenetic.nwb. And all our files will follow like a similar kind of naming schema. And then if you look at the actual file, we're viewing this in a software called HDF View, which I had mentioned earlier. If you want to get the same software, you'll be able to open any H5 NWB file with this and look at the information in a similar kind of visualization manner. And the link earlier in the slides will let you click there if you want to download this yourself. So we can look first at the acquisitions right here. And you can see we have eye tracking, raw running wheel voltage, rotation, running wheel supply voltage, and signal voltage. And we'll just open eye tracking to go through real quick. And you can see we have information on like the corneal reflective tracking, some eye tracking, some blinks, and some pupil tracking. So let's look at like the likely blinks real quick. You can open up your actual data here. and it will give you some information about what's stored in here. So we have conversion, which is just saying 1 because there's no specific units that are being converted here. Resolution, offset in whatever unit we're using. And then if we were to actually click here, we can look at our data and open it, and you can see it just mentions when the mouse is blinking or not. We're using 1s and 0s in this case, and these are just kind of like indexed across whatever unit we're using. And then if we were to look at the general, you can see we have some device information here. So if you were curious about like the probe we're using and stuff like that, that's stored here. We also have some extracellular EFIS information where you can look at some information related to the electrodes. We have just general metadata like our institution and the session ID and stuff. We also have some optogenetic information that you could look at, which stores stuff like what the excitation lambda is, the location of the genetic information, and that kind of thing. And then there's subject information. This could be really useful to actually filter files based on, and Carter will talk about some stuff like that later, where you could say, hey, I only want to look at mice that are sex, male or female, and you can find all that information here if that's relevant to your analysis. Additionally, there's these intervals, which are just showing which stimulus presentation was shown at that specific time. And this is actually related to another file we have here, another section of the file here, which says templates. So you can find a frame presented at one point. So you can look here, and you can see that we're showing frame 0 initially, and then we've switched to frame 17, for example, of this specific presentation. And if you're curious what that looks like, you can go here to these presentations and look at presentation 17. And if you click show data with options, you can actually see what the presentation looks like. And if you wanted, you could create a script that prints these out. So this is really helpful just to see like the actual stimulus, what is being presented at what time. And then, yeah, you could look at the black and white values if you want instead. And then in addition, we have the processing field right here, which has some opto tagging, running, and stimulus information. Again, you can look at these in more detail later, but it's mostly just data and timestamps for stuff like the running speed and that kind of thing. There's some specification information. This is really useful if you want to build custom NWB files. These are essentially extensions that let us store things in a specific way. So you can see we have one for the way we're storing eye tracking and extracellular ECFIS information. And then, yeah, we have the units table here if you'd like to look at stuff like the mean waveforms at a specific point, for example, or spike times, or the SNR, whether something has good quality or whether it's been marked as noise. All that information is stored here. And Carter's notebooks will go into a little more detail on what each of these things mean and how you can look at them in a little more detail in a programmatic way. All right, and then we have a optical physiology file right here. These are pretty similar, but they're a little different because obviously there's not as much spike information and stuff like that because it's 2 calcium. So again, there's an acquisition here, and this one's very similar where it just says eye tracking and then wheel information. And then there's a general. This one is again very similar. We have like a devices field, but instead of probes, we have information about the rig here. There's an optophysiology one. This one will have some information about imaging planes as well. And then there's the subject information. Again, this is the same type of stuff where it's just the sex, the species, the subject ID, stuff like that. And again, we have intervals. Now, this file, I'm not showing it on purpose because my laptop will run out of RAM if I do it. But instead of images, these ones have movies. So if you want, you can look at the movie you want to look at and then find the interval in the other section of the file here where we have the stimulus templates. And you'll be able to open it and actually look at what that frame of the movie looks like. It's stored as an image series. And then processing, we have very similar information, except we also have this one OFIS field, which will include some information about the average and max projection images, and then some OFIS motion correction information. Note that this is the non-raw OFIS file. If you're to look at the raw one, there's also like the actual raw movie attached here. Yeah, and then we have the same specifications one where we're extending the common NWB templates, or sorry, the NWB formats, and then the same stimulus information here. And yeah, that's more or less the demo on our NWB files and their schema. And now we can talk about DANDI. So DANDI is a platform for publishing, sharing, and processing neurophysiology data. It is very big in scale. Like there's over 500 terabytes of data and over 300 DANDI sets. And I think now it's at like 700 DANDI sets or something crazy like that. So this has gone huge. And you can share your files as easily as sharing a link, and you can generate a DOI as well if you'd like. So here's like a little example right here where you can look at, this is like a dendritic coupling one, although this is a embargo data set because they haven't done their full data release, but you could go simply at this mouse and see all the associated files. And DANDI sets are just these kind of data sets that are stored on DANDI that can be either open to the public or embargoed, and they're stored as drafts before official releases. So right now we've made 2 of them public, but they're still in draft form in case you find any file issues or something like that we want to correct. But eventually they will be changed into a full public release, and we'll have a DOI. But for now, you can just click on a link and it'll take you to the draft, and you can find the associated files with the mouse. And DANDI Hub is a powerful processing environment for analysis that Carter will talk about a little bit more. And just to give you a quick example, I'm going to go over DANDI really quickly, where we have DANDI Archive, and we could go to My DANDI Sets, and we could look at, for example, this is the dendritic coupling one that is being publicly released right now. You can see we have like this title, we have a bunch of keywords, contributors. You can find kind of a description of the set itself, and then if you wanted to look at the files, you can go here. They're separated by mouse. So you're like, I want to look at mouse 644972. Just click on that mouse, and you can find all the associated files. And if you wanted to download one, you would just click here. And if you wanted to share it, you just grab this link and send it to someone, and they can find all the files for themselves as well. All right. Oh, looks like it took me back to the first slide. Okay, cool. And then, okay, so let's say you want to find our NW files. You know they're on the DANDI sets, but you're like, well, I want to find a mouse that has specific metadata, and I don't want to code up something to find the mouse that has, for example, this genotype. We've created something called the data portal, which will help you find files, metadata, and notes for each session. So it allows you to filter sessions by all these common metadata fields, and then find the location of them on DANDI. So you can say, I want to find all the stuff associated with a specific mouse, click on it and find it, and this is for all 3 projects. Obviously, Glow, you won't be able to click on the DANDI links until February, but still, like, you'll be able to look at them for now. And they have some project-specific summaries and pictures, and I have a link here, and I'm going to, once again, show you the data portal. So this is what the data portal looks like. You can see we have this cool little picture, and then there's this front page that talks about the purpose of the website, how OpenScope works, some funding information. There's a nice video if you'd like to know some more background information. There's, if you want to look at the actual data sets, you can click right here. For example, if you wanted to look at Illusion, just click on this. It'll load because it's grabbing some information from our backend on the table. But once it has, you'll be able to see some information about the actual background of the project, information about the actual visual presentations, some experimental design information that might be helpful for you. And then you can find the actual table where you can filter by session. You can look at notes specific to an experiment. So for example, the session had an issue where the LFP data got corrupted, so it's missing that. Or this one has some slight drift on F that's been noted for. This one has a major drift. And if you wanted, you can filter. So you could say, I want to look at mouse right here. And you can find the session associated with that mouse. And yeah, that's more or less the data sets themselves. You can also click here to look at the notebooks associated with the session. And it'll load up, for example, some kind of HTML form. And you'll be able to read out the actual experiment and how you can access the data. You can also go to the actual notebook and run it and stuff like that, or we'll go into a little more information in the actual work. And then this is how the contact page if you'd like to something like audio. That's a little bit better, sorry. So anyway, if you'd like to reach out to us on Slack, there's a link right here. If you have questions, we can email link. We also have some GitHubs. I think we're having some minor internet issues.
Sorry, Hod, your audio is breaking up a little bit. We can hear some of it, but
Yeah. Is that better? I turned off my video.
Let's try one more time.
Yeah. Is
Yeah,
this
that's
a bit
better.
better? Okay, perfect. Yeah, I just turned off my video. Sorry about that. Yeah, so I'll just go over the contact information again really quickly. We have this Slack channel you can reach out to us on if you'd like to ask specific questions. We have an email if you prefer that. We also have a GitHub link for the data portal that just, I think, goes to my GitHub page, so you can send me questions or put a PR or something like that. And then if you want to contribute to the actual code, it's right here. And yeah, that's more or less it for the data.
Could you re-explain the notebook page? Because I think all of your word we're not going through.
Sorry, no worries. Here we go. So if you want to look at what are the notebooks associated with the session, all three of them are right here. We have the Illusion, the Global Oddball, and the Dendritic coupling project. You can just click on whichever one you'd like. So let's say Global Oddball for a second. It'll load in. This is just like an embedded HTML of one of Carter's notebooks, but you can read through it. It gives you some background information on the project. It'll tell you how to set up an environment, and it will show how you can actually access the data as well as the metadata for the table, like you can see which ones have which one sex and stuff like that, and how you can download the file. And then if you'd like, you can actually click here, and it will take you to the GitHub hosted version of the notebook, where you can run this and look at any updates that have been done to the file. Yep. And then let's explore. All right. Yeah, and so now you have NDB files, you know where to download them, you know how to filter them. What do you do now? So we've written this OpenScope Databook, mostly Carter. I helped a little bit. Jerome did a ton of work. Everybody has contributed, and it shows you how to actually access the notebooks, how to access the NWB files, how to do some analysis on them, how to go through the metadata and stuff like that. And Carter's going to give a little more information on the actual notebooks and talk about his work.
Thank you, Ahad. Again, please let us know if there's any questions. Carter's going to be up next talking about following up on that a bit more. So Carter, if you want to go ahead and share your screen. And again, if there's any audio issues, Carter will let you know, but we can always shut down your video if it's taking up too much bandwidth. All right. And Carter, we can see your, there we go.
Oh, apologize. My was muted. Okay, I'm on the same router as Ahad, but so hopefully I won't have the same problems. Can you guys hear me though?
Yes, we're still seeing your Zoom screen. We saw your slides for a second. There you go. And they're just not full
Perfect.
screen yet.
Okay. Oh, not back there. All right. Okay, we'll just go back. Everything good? Can you see it?
Looks great.
Okay. So I'm Carter. I'm on the same team as Ahad, and we work with Jerome on OpenScope. So as Ahad just went over, we put a lot of work into developing this pipeline to generate our NWB files in the NWB standard. And after putting in all that work, we have our experimental, we have our data collection all standardized. We have our file formatting all standardized. And but what we lacked is a lot of really any sort of standardized type of analysis or any reusable analysis code. So the state of affairs about a year and a half ago, before we started the data book, was that our files were packaged, and we have we have all three of our OpenScope teams collaborating with us, and we wanted to basically be able to provide them code that makes the data more accessible, maybe explain some things, and just kickstarts some basic analysis with our data. Like Ahad showed, NWB files are pretty flexible and very useful standards, but they also might be a little bit daunting to grasp at first, right? Maybe without any sort of guidance. And there's no centralized code that sort of explains this, or how to use the data to begin with. So that was the impetus for developing the Databook. We wanted a platform that mitigated these problems. And so these were the goals we set out to achieve. We wanted to be able to access the files directly from DANDI. We wanted to be able to educate users on what the files are, how to use them, and what the data even are, what types of data we include. And then we also wanted to have this platform be able to host the computational resources for analysis, as well as the code. And with all of those things, we'd want to be able to promote the reproducible output, create figures and such that can be shared, and ultimately expedite the process towards publishing and developing meaningful findings and disseminating our data. So we did that by developing the Databook, and it is a Jupyter book. So that was at the suggestion of Satra Ghosh, who works on DANDI, which is a really great idea. Essentially, it's a website, and it consists of a whole bunch of Jupyter books, or Jupyter notebooks, excuse me, and they all just have various kinds of code, but they all rely on using our files off of DANDI, our already packaged data. We have them organized into chapters based on the subject matter. It's a public website. It's hosted on GitHub for anybody to use. It's up pretty much all the time, and it's linked to our public GitHub repository. And the really neat thing about JupyterBook is you can launch the notebooks onto a number of different computing platforms. So you don't even have to host the computation yourself, which I'll explain more thoroughly later. So I think the best way to explain the Databook is just to demonstrate it. So I'd like to go through some of that. Can you guys see this? Okay, I'll take the silence as a yes.
Yes, sorry.
This is the GitHub page. And like I said, you can just find this by Googling, just OpenScope Databook. I wouldn't recommend just looking up Databook because there are other platforms with the same name. So OpenScope Databook, and you should be able to find this on GitHub. And this is the public page. We have a table of contents. Each of these are notebooks with different types of analysis. But the main event is this link here. You click on this, or it's down here, and it takes you to the website. So this is JupyterBook. If you're not familiar, it has these nice sections and a really clean layout. Hopefully, it's easy to parse. Our front page just explains the design behind the Databook and how to use it. And yeah, the gist. We have this authors list here at the top, which I think is pretty neat. It lists the authors based on number of commits. That's what these numbers are here. And something that's really cool, we added a Sphinx extension. The great thing about JupyterBook is it's really extensible with Sphinx. We added a Sphinx extension that dynamically generates this author list based on contributors on Git. So if you add a contribution of your own or any kind of analysis, then you automatically get added to this authors list when we redeploy. So to go through the content of the Databook, we have, firstly, with this basics chapter, it is just the idea. I mean, Ahad gave a great, gave us some good education on NWB files earlier, but if you forget or you want something maybe more specific to how to use it in code, we have these notebooks here that just explain how to download an NWB file, how to open it, how to actually look inside and interpret what's in there. So for instance, we have this reading an NWB notebook, and if you wanted to launch it, you could basically go to this little launch button right up here and click on one of these three or four, I guess, computational platforms to launch on. I would recommend this one, Jupyter Hub. Ahad mentioned this, but DANDI hosts their own Jupyter Hub, which is just a platform to run Jupyter notebooks, and for free, and it has really close access to the S3 buckets that the NWB files are actually stored in. So it has really quick access to the NWB files for downloading and streaming them. So that's the platform I would recommend using. And if you wanted to launch like that, you basically press this and you can pick which type of machine you want to run it on. I'll pick Tiny for the purposes of today. And it basically loads up. You do have to log in with your GitHub. But and actually, it also saves all of your data for you in hard storage, so it's not temporary. Some of you might be more familiar with Google Colab. You can also launch there if you just prefer that. Downside though is that I don't think it provides hard storage in the same way. So and it has maybe more limitations than Dandy Hub, but we also have Google Colab support. Continuing on with the chapters, we have our visualizing NWB file section. So this is really a bunch of notebooks that just explain, that actually dive into the data themselves instead of just the NWB files. NWB is really flexible, like we said, so there are many forms it can take, but these notebooks are specific to OpenScope's NWB files. We cover like neuropixel, electrophysiology, but we also have files with our 2-photon optophysiology, like you've seen in our previous 2 presentations. And so these just sort of explain how to retrieve that data from the file and produce some basic plots, like maybe explaining what they are, like LFP or the raw 2-photon movies that Jerome mentioned earlier. And then one of the most important chapters probably is this first order analysis. We have all of these that are starting or like jumping off points for like actually more in-depth analysis of the data. We have stuff like getting the receptive fields out of the files, like, classifying neurons or like cell segmentation, things like that. And ideally, any of these, you could basically right here, take the file URL from Dandy and paste it. And if it's the right type of file, you can run it on one of these platforms and get really good output, which I'll walk through some specific notebooks in a second. But yeah, so these are really good jumping off points for any of these types of analysis, and we're constantly expanding these. And then finally, the chapter we're working on mostly right now is adding to the higher order analysis section. These are notebooks, maybe that are more computationally intense, more abstract representations of data. So this one is using Zebra, which is a machine learning model developed by the Mathis Lab. So we worked with them to make this notebook that produces, you basically train a model and then it produces latent embeddings. Let me scroll down to show what the output might look like. But it produces these basically latent space embeddings of I think this is a 2-photon file that it uses. And then we also have TCA, tensor decomposition as well. And like I said, we're expanding this and working on collaborating with other package developers to add higher order analysis. I'll go through these 3 notebooks in just a second. These are the data sets we're releasing today. We have a chapter of, these are notebooks for analyzing files that are as of yet still embargoed, but they should be released. The rest of the dendritic coupling data set should be released next month. I don't know if we already said that. And that's what these ones operate on. And then we have a method section that explains the internal implementation of the data book if you're interested, and then a bibliography and FAQ. And if you're not familiar with Jupyter Book, like this by bibliography is really cool because it gets dynamically generated by the citations you include in text, which is really nice. So those are the chapters. And now I would like to go through these 3 notebooks. So we made one notebook for each of the data sets we're releasing today to help people get started on analyzing it. And actually first, I'm going to go over dendritic coupling because that was what was presented first. So you already heard Jay's great presentation about this data set. It's 2-photon imaging. So all of our notebooks start off with environment setup. We've done our best to make sure that all the notebooks on the data book run on any environment, but it feels like a constant battle. But ideally, I think right now it's in a good position, and all of our notebooks do run on the various launching platforms with just the push of a button, so you don't have to worry about setting up an environment. This next section here is similar to the data portal Ahad just showed, but basically it's a table basically listing the files in our data set. So it's a little less thorough than Ahad's data portal, so I'd recommend going there if you want something more thorough, but for a nice notebook view, you can see these are our files. comes with the path and various things like the experiment ID, imaging depth, location, etc. And we have a little summary here of the data set. So the current release of the dendritic coupling data set is just 2 mice, which at 6 sessions, I believe, per mouse and 8 imaging planes per session, that's 96 files. So next you could just enter the URL of the file you're interested in, and it would be downloaded through the DANDI API here. You don't have to have it pre-downloaded. And then you can view the sort of metadata about the imaging plane you're looking at. So this one is, the imaging depth is 144 here, and it's viz PM is the location. And Jay did a great explanation of their stimulus, but this also sort of shows them. So you have each of the sets of movies. These ones are the forward ones, and then these are reverse. These are forward reverse, and these are reverse forward. These are just screenshots that are displayed here, pulled from the file. But if you ran this notebook, you could also actually play the stimulus movies if you wanted to get a good idea of what they look like. And then let's see, next you can also pull the stimulus times from our NWB files. So this is the stimulus table. It might look kind of daunting, but it's basically just a list of what's shown at every frame throughout the session, and like what type of stimulus is shown. So my code here just selects all of the times where frame 190 of the movies, of this particular movie, was shown. And that's because this is when the movie reverses playback. So this is when the unexpected stimulus event happens. And we also can extract the fluorescence trace for all of the all the ROIs from the file here. And if you wanted a better understanding of how the session is laid out, this pulls all of the different stimulus epochs from the file and creates this nice plot. So this just shows you what the timeline of the session actually looks like. And you can see the random sequence of movies shown in this file. And you could zoom into this if you wanted a better idea at a certain point, followed by the rotating gabors and also the fixed gabors at the end. An additional note is that our files all come with, all the OFISC files come with behavioral data. So in this case, we were showing the eye trace of the mouse over time. We also have the running speed of the mouse on the wheel throughout the duration of the session. So you could, I mean, this is pretty messy, but you could basically dissect this a little bit more and do something with mouse attention or other behavioral queries you might have. But the meat of the analysis for this notebook is taking those the fluorescence and the stimulus times, and you can basically align them in time and create a 3-dimensional matrix that I believe is the 3 dimensions are cells, trials, and time. And out pop these plots. So this is just a really nice view of how the ROIs behave across trials and through time when given the stimulus. If I zoomed in here, you can see the line that marks the onset of the stimulus. And I also have some code to select responsive cells. This one is just it's just a paired t-test. As you can see, only 2 cells are selected. But maybe you could change this to develop a more robust inclusion criteria or modify some of the parameters. And finally, for this file, I think Jerome alluded to this earlier, but we include the raw movies. So for every main file in this data set, we also include a copy of that file that also has the raw movie taken from the imaging plane in the brain. The reason we don't just include that in all of our files is because it's really big. So this is specifically if you're interested in looking at the movies. The download would take some extra time, but this section of the notebook, basically you can download it and then you can actually play and see the movie that's taken from the brain here if you ran this notebook. So that's an overview of the dendritic coupling notebook we have. Next, I'm going to go over global local oddball, which once again Jake gave a great explanation of, so I'm not going to read all of this. And here's like an example of the local oddballs and the global oddballs that Jake explained. We have the same sort of environment setup and a similar breakdown for the the subjects, the whole data set. So here we have 14 files. That's one for each mouse. You have the breakdown of the sex and the genotype. And we can pick one from the table and download it. And so our NeuroPixel datasets are a little bit different than our 2-photon datasets. They rely on a totally different technique, right? So we have these probes here. This plot was actually generated from a different notebook, but copied here. And you can see it actually it's really great. It gives you actually just a display of the probe tracks through the brain. You have 6 probes, and you can see where they go relative to each other. You can see there's kind of this wavy shape that they seem to have. That is not from the real trajectory of the probes. When we take the coordinate framework that we use for the probes, and then we take it from the real mouse and map it onto this model mouse that we're rendering it in, the coordinate space gets a little bit warped. So the probe tracks get a little bit warped. So it's not perfect, but it does have a pretty good, gives you a pretty good idea of what the setup is here. So with that, you can look, these are all of the, this is a table displaying all of the neuronal units in the file. And these are all the different sort of quality metrics that Jake mentioned in his presentation. And most importantly, there are the spike trains for each unit here. So really easily, you can basically view all of the regions that basically were detected by our probes from this units table. And what I do is I select units from the table that have a good quality, so units that aren't noisy, and I select them from these regions here, which results in 175 selected units. One more thing about the experimental design that we can plot is the session timeline. Like I already showed for dendritic coupling, we have something similar here, but this shows the spiking behavior of the selected units throughout the duration of the session. You can see there's the regular gradings followed by intermittent gradings. And then at the end, we have that optogenetic stimulation period that Jake mentioned. We also have this receptive field mapping presentation epoch here. As you can see, there's sort of overlap between this section and the gradings presentations. That is a mistake in the stimulus. So we were actually able to see that and catch it from this plot here. So it shows you how useful these are. But I would recommend caution if you're analyzing this portion of the session because the two stimuli are conflated. Let's see. Next up, we can take the stimulus times from our stimulus table, just like I mentioned in the previous one. And what I select here is the local oddball stim times and the global oddball stim times, or at least some version of those. So I select a certain number of each of them. And now that we have our unit spikes and our stimulus times, we can do the same thing we did in the last one, but for spiking behavior rather than fluorescence. You can make a 3D matrix, and we can plot with these plotting functions and really easily pop out these. So for any of the global local oddball files in the data set, you could just change the URL up there and rerun it, and you would get these great response plots. And we also use a metric to filter out spells that are specifically responsive. Just like what confirms what Jake seemed to have had concluded that the local oddball responses do seem to be a little bit more numerous and a little bit clearer than the global oddball responses here. So that's good news. And finally, we include this opto tagging section, which basically does the same thing, but instead of selecting stim times based on the exact type of stimulus shown on the screen, it's the type of laser shown. So, but it's essentially the same type of analysis. You could change these parameters here if you wanted and customize this analysis, but you get a similar type of thing where you can see units and their responses to the laser stimulation. So finally, I would like to, for these notebooks anyway, I would like to go over the notebook we made for illusion, which Hee-Young will be presenting shortly. So I won't explain all the details of the experiment. I'm sure she has that ready, but I will just briefly mention the type of stimulus shown on the screen. So we have these, well, I'll do that in just a second. We have our environment set up, We have our experiment summary, and we can pick a file from that after seeing this summary here. And this data set also includes in our files the stimulus templates. So Ahad showed these briefly also. These are the stimuli frames shown for the illusion experiment. You can see that some of these cells have, or excuse me, some of these frames have like illusory contours, like these imaginary rectangles here, and some have real contours, and some have neither, and various permutations of these shapes here. And then we also have the receptive field mapping, and then these size stimulus templates here. So you can just view those from the file. We have the same setup for the probes. This is the exact same figure. And we have the unit spikes once again. So this notebook is pretty similar. We select units based on the same criteria. And then we have the timeline. The timeline for illusion looks a bit different, but it's basically various, these epochs are the different these different sections of stimulus or templates. And this is the firing rate over time to get an idea there. And then after selecting, in this case, we're selecting the stimulus times when frame 1 is shown and the stimulus times when frame 0 is shown. So that's when a frame with illusory contours occurs versus a frame without illusory contours. And after doing the same thing, we get these, and we can filter them down between we have the illusory contour frame responses and then the ones that don't have any contours. And we can select them with the same inclusion criteria. You can modify this to make it more robust, like I said. And in addition, this notebook also has the receptive fields. So we have that receptive field mapping section of the session timeline, and these are in this particular data set. These are where the receptive field mapping presentations occur on the screen. And with this code, we can just tally up the spike responses to these receptive field figures for a number of units. And finally, just like the last one, we also have opto-tagging where we can do the same thing and get receptive fields for them as well. And you can see there's some pretty clear behavior here following the onset of the laser. So that's good news. So these are basically designed to help people get into analyzing the 3 data sets being released today. And pretty much everything in these 3 notebooks are covered in more detail and more thoroughly in the rest of these notebooks for people who are interested in checking them out. And then there's also a number of other analyses that I did not cover here that are shown in the data book. So I'd highly recommend using this platform, especially if you want to analyze OpenScope data, but probably many other NWB data sets as well. Let me get back to my presentation here. Okay, so I want to talk about the outlook of our project. So we spent a year and a half working on this, and we intend to continue working on expanding the types of analyses, expanding the different types of, or the different projects that we're working on with this. And we'd like to have this project evolve to fulfill needs as they become clear and as OpenScope grows. And as open science as an ecosystem grows, we want to start incorporating elements from other packages, other neuroscience analysis packages, and have many collaborators start working on this. We demonstrate like review with our GitHub pull requests, and ultimately we want this our platform to help maximize the impact of our data and open science as a whole. To conclude, I just want to say that OpenScope and the data book in particular has had a lot of people contributing to it. We have internal as well as our many external collaborators working on it, and the project definitely couldn't be done without the efforts of all these people. And they're doing a great job at working together to promote open science. So I will pass it off to Jerome now.
Yeah, let me turn my video.
Yeah.
I just want to emphasize that we put a lot of intent, as you can see, a lot of work to sort of facilitate data reuse, and it's really important that as a community we we tried to build a really collaborative environment around this really open door approach. And so we shared a link to Slack. Please join this group and try to work with us. Come be part of it, and let's not try to make this a competitive endeavor. This is really important for the team and for the field as a whole. As we build this observatory platform, we want to set rules that are like really respectful of everyone's work. But that being said, I'm going to pass it on to Yi-Young.
Hello, everyone. Everyone can hear me, right?
I can hear you. Looks good.
Awesome. Okay, I'd like to thank the OpenScope team for organizing this wonderful webinar, especially Jerome, and also for the entire OpenScope team for being truly wonderful collaborators and for pioneering this platform, trailblazing the way for open science. My name is Hae-Young Shin. I'm an assistant professor at Seoul National University in South Korea. But this work was started when I was a postdoc in Dr. Hilal Adesnik's lab at UC Berkeley. And today I'm thrilled to share with you my research and to introduce you to the Illusion OpenScope dataset. We are currently working on a paper on sensory inference where the Illusion OpenScope dataset will be introduced for the first time. And the link to that paper is right here. All right. So the outline of my talk is as follows. There will be 3 sections. First, I'll motivate why we are studying illusions, especially illusory contours. And then I'll introduce you to some of the cool science that we did with the OpenScope dataset, which again is described in far more detail in this bioRxiv preprint. And lastly, I'll spend a couple of minutes introducing the nuts and bolts of the dataset in the hopes of making it easier for folks wanting to try their hands in analyzing this open source dataset. So neuroscientists have spent a lot of time, the last several decades, in fact, studying the famous grading stimulus, which look like these. And neuroscientists that feel bold might even venture to ask how well neurons in the visual cortex can discriminate between this stimulus and this stimulus, which is tilted 1 degree relative to the left stimulus. But accurate orientation discrimination is really not what our brain was built to do, and frankly, we are not very good at it either. So what was our brain built to do? Our brains are really good at doing perceptual inference. That is, our brains have evolved to infer objects from incomplete or ambiguous sensory information. For example, this cat has to be able to infer that there is a mouse in this hole, given this incomplete outline of the mouse. So what I believe we should all be looking at is not how the brain can distinguish between these 2 oriented grading stimuli, but to study how the brain can infer this from that stimulus. So we are not the first people to get excited about illusory contours. Primate neurophysiologists such as von der Heydt and colleagues, as well as Lee and Nguyen, really pioneered our understanding of how illusory contours are encoded across the primate visual hierarchy. And they found that top-down feedback from V2 to V1 is a key component of this mechanism, a finding that is also highlighted in this Wikipedia schematic of the visual hierarchy. So we wanted to study the neural mechanisms of illusory contour encoding in mice, first because we are only beginning to understand the mouse visual hierarchy, and utilizing the illusory contours will contribute to our understanding of the mouse visual hierarchy. But more importantly, we wanted to dissect this computational circuit motif right here using single-cell resolution neural manipulation techniques such as two-photon holographic optogenetics, which we could do with mice, but not yet with primates. For a mouse to be a meaningful model organism for investigating illusory contour inference, we first need to know whether mice can perceive illusory contours. Fortunately for us, at least 2 prior studies have demonstrated that mice can indeed perceive illusory contours and they experience the illusion of the illusory contour. Here, I'm showing figures from the Okuyama, Uchimura, and Komai paper, where they train the mice to perform an orientation discrimination task, vertical versus horizontal, using real bars. And then they test whether the mice can transfer their performance to illusory contours. And they showed successful transfer of the behavioral performance without additional training to illusory contours, which demonstrates that mice can perceive the orientation of the illusory contour. That is, they can perceive the illusory contours themselves. So in our study, we focused on gathering high-throughput neural data while the mice were passively viewing illusory contour images. The OpenScope team at the Allen Institute collected the NeuroPixels dataset, and we in the Adesink lab collected a parallel dataset, a massive 2-photon imaging dataset that we plan to release as well, publicly release as well in the future. So that's something to look forward to. So our illusory contour images look like this, and it was inspired from the behavioral studies that I described earlier. Now, to see whether illusory contour images evoke a gestalt response, where the perceived whole is greater than the sum of parts, we designed the LC image pair by mixing and matching the components of the IC image pair. So what I mean by that is that we took the top half of the IC1 image and then the bottom half of the IC2 image to get the LC2 image, and vice versa for LC1 image. When we look at the response of individual neurons to this set of 4 images, we find that there's a sparse subset of neurons that respond to one of the IC images, but not to either of the LC images. So we call these neurons IC encoders, and this means that these IC encoders respond emergently to the specific combination of the inducer segments that make up the illusory contour image. Further, we observed that these illusory contour encoders are tuned to the orientation of the illusory contour. So the IC1 image contains an illusory contour that is oriented in the 135-degree direction, and we see that these neurons are indeed tuned to that orientation. And since orientation is an emergent feature of the illusory contour, we can conclude that these illusory contour encoders are indeed responding emergently to the illusory contour. And we can find these neurons across the visual hierarchy, but really the presence of these neurons neither confirms nor denies the existence of the inference signal in each brain region. So we wanted to come up with a way to test whether each brain region contains the inference signal of the illusory contour. So to look at the neural representation more comprehensively, we wanted to leverage a decoding approach where we decode the visual label from the trial-by-trial neural data. And not surprisingly, the IC and LC images evoke distinct patterns of neural activity in the visual cortex, which can be confirmed via this cross-validated performance of the neural decoder. But how do we use this decoder to examine inference representation? To do this, we devised a clever trick where we designed an image that contains a real edge, but has equal pixel overlap with the IC image and the LC image. So we call this the TRE image, for short for T-shaped real edge images. And to show you what I mean by equal overlap, I'll show you these images in sequence. So here is our IC1 image, and this is our TRE1 image. And we can see that the bottom left portions of the two images overlap. And then between the TRE1 image and the LC1 image, the bottom right portions of the images overlap. And so if you quantify the number of pixels that overlap between TRE1 image and IC1 image, and TRE1 image and LC1 image, then those numbers are exactly identical. So there's equal overlap between TRE1 and IC1 and TRE1 and LC1. And we can do the same thing for the other set of IC and LC images and make TRE2. Now, having designed these images in this way, we can use the decoder that we trained to discriminate between IC and LC evoked neural activity and make it decode the TRE evoked activity. And what we can expect from this analysis is that if the neural representation is strictly faithful to the images themselves, the decoder would trivially reflect the amount of overlap between the images. So the TRE will be equally likely to be decoded as the IC and the LC image. On the contrary, if the IC image evokes the representation of the illusory contour, that inference signal will be similar to the representation of the real edge contained in the TRE image. And therefore, that would endow a higher similarity between the TRE1 image and the IC1 image, which is an information that is not contained within the pixels themselves. So therefore, the TRE would be more likely to be decoded as the IC image than the LC image. And what we see when we decode neurons in mouse V1 recorded in the NeuroPixels data set is that there is an overwhelming evidence for inference signal, the presence of inference signal in mouse V1. And when we apply this analysis across areas, we see strong inference signals in V1, LM, and AL. And furthermore, if we look at the same thing in our two-photon imaging data set, we get a remarkably consistent result, which is that only V1 and LM contain inference signals, and the other areas contain less of an inference signal. And interestingly, we also find that if we just look at the layer 4 neurons of mouse visual cortex, then we no longer get a significant inference decoding performance. And what this implies is that layer 4 is more faithful to the sensory information contained in the images themselves. And this makes sense because layer 4 is the input layer, and it is thought that sensory information is more faithful in the lower parts of the visual hierarchy, which layer 4 is lower than layer 2, 3. Okay, so we won't have time to get into more detail, but in the rest of the paper, we use two-photon holographic optogenetics to show that V1 IC encoders drive recurrent pattern completion, indicating that the IC encoding model should be updated to include these red arrows from V1 IC encoders to segment responders. This suggests that V1 IC encoders play an active role in shaping IC encoding, contrary to the previous model, which assumes that all the interesting inference computation happens in higher visual areas, and V1 neurons passively reflect whether the inference computation in higher visual areas was successful or not. And more generally, this result implies that the recurrent looped connectivity that spans multiple stages of the visual hierarchy is critical for illusory contour inference. So this is a critical step forward from just saying that feedback activity is important. It also highlights the importance of the recurrent connectivity that spans multiple areas. So I'd like to use the remainder of my talk to introduce the data set in more detail, particularly the NeuroPixels OpenScope data set that is being released today. So in this data set, there were 6 visual blocks, as Carter explained. And 4 of the blocks, visual blocks, are the IC blocks. And then there's 2 blocks in the ladder, each indented for receptive field mapping. That's the RFCI block. And then size tuning mapping, which is the size CI block. And the 4 IC blocks differ in black and white color inversion between the first and the third, and the second and the fourth, and also orientation. So ICWCFG1 is rotated 45 degrees counterclockwise from ICWCFG1, and the same is true for ICKCFG0 relative to ICKCFG1. And then after the 6 visual blocks, there's the opto tagging session that lasted 27 minutes. So these are the images that were presented in the ICWCFG1 block. By the way, ICWCFG1 is short for Illusory Contour White Configuration 1 block, meaning white is like the color of the inducing circles, and then 1 is just the orientation. It refers to the orientation of these circles. And to help your analysis, I am indicating the Python index of each image contained in the NWB files in the parentheses here. And just to provide a snippet of what it looks like, what the mouse sees during the experiment, I'll show you a short video clip. So this is what the mouse sees during the experiment. The 4 white circles stay in place in the intertrial intervals, and each stimulus is presented for 400 milliseconds, and the intertrial interval is also 400 milliseconds. And in addition, the 4 white circles are presented for 24 seconds at the beginning and the end of this block. And I'll briefly show you the images for the other illusory contour blocks. This is just rotated counterclockwise 45 degrees from ICWCFG1 block. This is black and white inverted from the ICWCFG1 block. And then this, again, is rotated counterclockwise 45 degrees from ICKCFG1 block. That's the ICKCFG0 block. And then, as I mentioned, the latter 2 blocks are intended for receptive field mapping. That's the RFCI block. And then the size CI block is intended for size tuning mapping. And in the RFCI block, circular patches of drifting gratings were presented in 9 different locations. And these gratings were presented either as classic gratings, where the gratings are within the circular patches, or inverse gratings, which are essentially full screen gratings that are occluded by a gray occluding circular patch. And in the size CI block, the size of these center gratings were varied between 6 different sizes. So right now I'm going to show you a snippet of the RFCI block. As you can see, the drifting gratings spin in 0, 45, 90, and then 135-degree order, while the circular patches stay at each position. This is to activate all neurons that have classic or inverse receptive fields at each location, regardless of the direction that each neuron is tuned to. And each drifting grating at each direction is drifting for 0.25 seconds with 0-second intertrial interval. And the spatial frequency of the drifting gratings for both the RFCI block and the size CI block are fixed to 0.04 cycles per degree, and the temporal frequency is fixed to 2 cycles per second. In the size CI block, both the direction and the size were randomized on each trial, and each drifting grating was presented for 0.25 seconds, followed by 0.5 seconds of the intertrial interval. And lastly, the optotagging block was inserted to identify parvalbumin and somatostatin positive inhibitory interneurons in respective transgenic mice. We had 12 different optogenetic trial types, each repeated 50 times, so 600 trials in total, and this block lasted about 27 minutes. So that in a nutshell is the data set, and I wish you a very happy data mining. And of course, feel free to reach out to us to share with you, share with us what you find, or if you're interested in collaborating with us. And I'd like to thank Hillel, who mentored me throughout this project, Mora, Lamia, and Uday, former lab mates of mine that I collaborated very heavily with. And of course, I'd like to thank Jerome, most of all, for heading the OpenScope collaborative platform. And lastly, I'd like to thank my budding emerging lab, especially Yaren Kim, who is a talented graduate student who helped me clean up some of the code for the data release. And with that, I'd love to take your questions.
Thank you, Young.
Thank you.
It was a wonderful talk. I have a similar question that I ask Andre and Alex.
Okay.
So you show this beautiful decoding analysis and the importance of recurrence in your hypothesis. And Andre and Jake and Alex spoke a lot about the delayed response of the global oddball response. You know, so it's probably involved feedback. And so I'm curious if this is also an avenue of overlap between the project. Have you considered looking at longer timescale response, and could that be done across data set?
That's a very thought-stimulating question. I have to think a little bit more about that. I think that there are like generalizable principles, especially in the inference connectivity motif that we kind of characterized and discovered. And it would be amazing to see if that circuit motif generalizes across different inference computations. And so in that regard, I am very interested in looking at other data sets. I'll have to think a little more about specifically with regards to those two projects, how it can be applied. But yeah,
yes.
that's...
And are there Alex and Andrew mentioned subcortical areas, and have you done analysis of hippocampal data, or is that something that you're working on? I'm curious.
So I did look at LGN just to see if it's more faithful than layer 4, for example. There weren't very many neurons there, and it wasn't consistently targeted, so that's not surprising. I also wanted to look at LP for a similar reason, the higher order thalamic nucleus for visual processing. So up till now, I just focused on thalamus for subcortical analysis and didn't get much there. I think, yeah, hippocampus would be potentially very interesting to look at maybe reactivation or like, recall during spontaneous periods and stuff like that. Yeah.
Okay. What's the best way to reach you out for people that want to collaborate?
Yeah, email. So I have the email in the last slide of my talk. And yeah, you can Google me and find my website. That would be great. Yeah.
Okay. Are there questions from the audience, Carly?
No, we're all set for now. And then just a reminder, this will be recorded and on YouTube later if people want to go back and watch and share it. And always, I think that everyone had contact information if people do want to reach out to the presenters or in the Slack channel if they do have follow-up questions.
I do want, before we leave, I want to thank Maddie, Macarena, and Carly for helping organize this session. I took a lot of coordination because we and also for all the speakers that some of them woke up early and some work late to make this happen, because we are truly international collaboration. And so I'm really thankful for the effort.
Thank you, everyone, for your help coordinating time zones. And with that, I think we can close it out today, and we'll talk to Maddie about getting it posted for reference in the future. Thank you, everyone.